In this video, we are going to investigate the abundant and very important receptor tyrosine kinase, or RTK. The learning objectives for this video are to become familiar with the role of kinase activity in RTKs and to appreciate the role of RTKs in normal physiology and in cancer. Receptor tyrosine kinases, or RTKs, are a large family of receptors, some of which you may have heard about in your biology classes. These include epidural growth factor receptors, or EGFRs, insulin receptors, vascular endothelial growth factors, or VEGF receptors, and platelet-derived growth factor, or PDGF receptors. RTKs are critical for the control and regulation of essential cellular processes including cell cycle progression, proliferation, differentiation, cell metabolism, including glucose uptake, and overall cell survival. Since they are such a key player in cell survival, mutations in RTKs have been discovered in many cancer types. About 20% of breast cancer patients have tumors that overexpress RTK, and among the 58 known RTKs, 30 of them have been implicated in oncogenesis or the formation of cancer. All known RTKs are monomers in the cell membrane. When a ligand binds each monomer on the extracellular domain, the two monomer domains create a dimer. This action brings the kinase domains of each monomer in close proximity to each other and allows for autophosphorylation of the receptor. The phosphorylated dimer is then ready to activate intracellular proteins to trigger a signaling pathway. The exception to this is insulin receptors, which are disulfide-linked dimers, and insulin binding to the extracellular domain leads to increased autophosphorylation to the cytoplasmic domain. It is possible for two ligands to create a dimer and then together bind two monomers or other ligand complexes to initiate receptor dimerization. For the purposes of this class, this diversity in RTK activation is brought up so you can start to appreciate 1. How complicated RTK activation can be, and 2. The body has many mechanisms to regulate and control RTKs. This creates a challenge when trying to treat patients with abnormal RTK activation due to a mutation or toxin, since the treatment needs to reverse the specific type of activation. It can be very difficult to determine what part of the RTK signaling pathway, either the ligand, the receptor, or downstream signaling, is abnormal and why. Cells have many types of receptors on their membranes which can work in concert with each other. Here is an example of where activation of RTK or GPCR leads to PIP3 activation and the regulation of AKT. AKT plays many roles, including the inhibition of inhibitors, which control many cell functions we already discussed in relation to RTKs, including cell proliferation and survival. Due to RTK's role in regulating AKT, RTKs are a key player in many types of cancer. Excess ligand or ligand-independent activation of RTKs results in the kinase domain constituently on, and there can be an increase in proliferation, differentiation, invasion, metastasis, angiogenesis, cell survival, and cell cycle progression, all of which can result in cancer. Here are two illustrated examples of drugs that can be used to try to reduce RTK activity. A small molecule inhibitor, tyrosine kinase inhibitors, also called TKIs, can inhibit the tyrosine phosphorylation of the receptor, while an antibody, a biologic, can bind the ligand binding sites, force the two kinase domains apart, and stop autophosphorylation activity, and therefore stop downstream signaling. Small molecule inhibitors and biologics are being extensively developed for drug use and are on the rise for FDA approval. An example of a physiological function of RTKs is demonstrated here with the insulin receptor. After a meal, and therefore an increase in glucose uptake in the blood, the peptide hormone insulin is released by the pancreas into the bloodstream. Binding of insulin to the insulin receptor results in an increase in autophosphorylation and the activation of downstream signaling. This signal leads to AKT activation and an increased uptake of glucose by the glucose transporter. With insulin resistance, the body does not respond correctly to insulin, which may be due to a mutation in the insulin receptor. Research is currently being done to better understand the complex nature of insulin resistance and how it contributes to diabetes. In summary, RTKs are monomers with an extracellular domain, which binds a ligand, a transmembrane domain, and an intracellular domain with intrinsic kinase activity. When activated, the RTK monomers form dimers, and the intracellular tyrosine kinase activity causes autophosphorylation. One of the key downstream proteins of RTK signaling is AKT, which helps regulate cell cycle progression, proliferation, differentiation, cell metabolism including glucose uptake, and overall cell survival. This is part of the reason why overexpression of RTKs in abnormal cells leads to cancer.